Welcome, everyone. We'll wait just a second to let everyone enter the Zoom. Okay. Again, welcome, everyone. My name is Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. This webinar, like our series, is presented by ASCCT and the European Society for Toxicology in Vitro. And I want to encourage you to visit our websites to find out more about our organizations and consider becoming a member to support programs like this webinar series, the awards we provide to students and early career scientists, and other activities to engage the in vitro and computational toxicology communities. You can also join our mailing lists for announcements. The webinar is recorded, and the recording will be posted on the ASCCT website a few days from now. In the webinar platform, you can ask questions of the presenters using the Q&A feature at the toolbar at the bottom of your screen. And we'll have a Q&A period after each presentation today. Add your questions at any time, and the other participants can see questions and vote for them by giving them a thumbs up. We've also enabled closed captions, and you can turn them on or off in the toolbar. And finally, please say hello and let us know where you're joining from. We're really happy you're here today. Let me give you a couple of brief announcements. First of all, ESTIV's 23rd Congress will be in Maastricht in 2026 this year in June. There is still an opportunity to get involved. They have a last-minute abstract deadline for posters open until the 8th of May, and early bird registration ends April 12th. So head on over to their website and figure out if you are able to join them. We will also be having the 15th annual ASCCT meeting in Research Triangle Park, North Carolina. We are almost confirmed, but look like the dates are going to be October 28th through the 30th. Plus, we'll have a Society for Adverse Outcome Pathway ancillary meeting on October 27th, the day before. And session and CE proposals are open. Those are due on May the 1st. So come on over to our website and find out more about that as well. Finally, the Physicians Committee for Responsible Medicine in Johns Hopkins is presenting a summer immersion on innovative approaches in science. This covers all of toxicology and biomedical science in June in Baltimore, and there's still an opportunity to apply and to apply for some travel support opportunities. So head on over to that website to find out more about that. In terms of our webinar series, visit our website to see all of the society events as well as partner events on that website. If you are an ASCCT partner and you want to promote an event, be sure to let us know. But also we have the next webinar coming up April 30th, and that will feature Dr. Sean McCullough and Dr. Sizan Aydin. And you can register for that webinar on our website. Today, we have award winner presentations from our last annual meeting. These folks won awards from the Society. So the first presentation is Nyssa Tucker. She will be presenting on RADISH ROBOKOP Assisted Discovery of in silico Hypotheses, a case study of mechanism linking nuclear receptors to liver disease. She is a travel award recipient from the annual meeting and is also a fifth-year computational toxicology PhD candidate at UNC Chapel Hill. Along with the research interests embedded in today's talk, Nyssa is also passionate about building strong networks between people towards a resilient, community-focused system of collaboration addressing pressing social concerns. Nyssa is a member of the Society of Toxicology, Local UE 150, and UNC's Molecular Modeling Laboratory. Our second presenter today is Dr. Ivo Djidrovski. He is a computational toxicologist and AI research lead within the Virtual Human Platform for Safety Assessment Project at Utrecht University. His work focuses on developing auditable, interoperable AI-assisted workflows to support non-animal chemical safety assessment and next-generation risk assessment. He leads the development of OQT, an AI-assisted orchestration layer for the OECD QSAR toolbox that formalizes read-across into structured, policy-aware execution traces. His research further explores federated orchestration architectures like ToxMCP that enable transparent integration of read-across mechanistic pathway knowledge and PBK modeling. In parallel, he founded Infor.AI to translate agentic AI infrastructure into real-world industrial toxicology applications. Ivo Djidrovski is a 14th annual meeting Ray Tice Tox 21 Student Award winner, presenting augmenting chemical hazards and risk assessment through large language models and AI agents. Our third presenter is unfortunately not going to be able to join us today. Amir Daghighi is a student studying here in the US. He's from Iran, and as you can hopefully understand right now, with the war going on, he's not able to focus on his presentation. We hope that you will keep him in your thoughts. We hope that his family is safe and hope that Amir is able to get the information he needs. We will welcome Amir to present with us another time. Okay, now I'm going to turn the floor over to Nyssa. So please go ahead and share your screen.
Well,
everything looks good.
Great. Okay. So hi, my name is Nyssa. Excited to be here sharing this work today. This is part of my dissertation work in a project that I call RADISH, or ROBOKOP Assisted Discovery of in silico Hypotheses. So I'm looking at finding new mechanisms to link nuclear receptors to liver disease, and specifically I'm looking at fatty liver. And yeah, I'm in my 5th year of a PhD at UNC Chapel Hill. I have no conflicts to declare. This talk I planned for 15 minutes, and I'll start by covering my disease of interest, so fatty liver disease, before discussing the reference that I'm going to be using, or the AOP wiki, looking at fatty liver disease entries. Then I'll introduce the concept of a knowledge graph, some specifics about the biomedical knowledge graph that I'm using here, ROBOKOP, before focusing on the validation strategy to make sure then that ROBOKOP makes sense to use to nominate some mechanistic pathways. And I don't want to hold anyone in suspense. These are the 3 genres of pathways that I think are important kind of at the, and then we'll get to why I think that at the end. So as I mentioned, this is part of my dissertation research. Effectively, I'm using this knowledge graph tool to, of course, look at the overlap with adverse outcome pathway wiki entries for fatty liver, but I also want to identify some new mechanistic features to support my overall research goals, which is to predict complex biological disease, fatty liver disease, using screening endpoints. So AOPs, if we deconstruct them, could be or are genes that we can measure in screening paradigms, and I'm using those to build individual QSAR models, quantitative structure activity relationship models, and then integrate those to make an overall prediction of a hazard. So the point of this project is to nominate additional mechanistic support that I could include in that predictive toxicity estimation. So again, as I just said, I'm looking at showing for y'all today the development of this protocol to use knowledge graphs and identify new mechanisms. And here I reiterate the three mechanisms that I found most interesting in this work, which is lipid response, oxidative stress and response, and adipogenesis regulation. So fatty liver disease, why should we care about it? Well, first of all, the third of us, or a third of the people who consume a Western diet are likely to develop fatty liver, either through a genetic contribution, psychosocial factors like stress or community support, eating behaviors, exercise behaviors. And then the one I'm most interested in as a informaticist is chemical exposure. So of course, this Western diet as a feature of the fat compositions of our foods, but also this disaster and occupational exposure is really interesting to me. So vinyl chloride is a prototypical stressor for causing fatty liver. And is evidenced in the occupational studies linking workers who work with polyvinyl chloride, making like PVC pipes. But also, you could eat perfectly and have perfect genetics and have perfect eating patterns. But if you lived in East Palestine, Ohio, for instance, and a train derails next to you with a bunch of polyvinyl chloride, this may contribute to your development of something such as fatty liver disease. This is a 2014 paper where the authors are looking at rodent models, where on the left we have a control diet, on the right is a high-fat diet. And this first row looks at visualizing fat in liver cells. So these are liver slices with an oil red O stain. And what we can pretty much immediately appreciate is there's a lot of red showing up here. And so if we zoom in on that with an H&E stain, and look at the surrounding tissue structure difference, I hope we can all appreciate that there's alterations in the way these cells look. So they're phenotypically going to be different. All of the organelles are pushed out to the edges of the cells and kind of squished. So what we can see here is a change in the form, but also a necessary, or we can infer that there's going to be a change in the function of these cells too, just based off of the extreme change that we're seeing here. So fatty liver could be considered a possible susceptibility factor for people, as the liver is the major site for detoxifying compounds. And although initially a simple steatosis or a simple fatty liver is both asymptomatic and reversible, if the same pressures of the environment or chemical exposures continue, these structures may stop being as flexible. So there may be some rigidity forming, fibrosis, et cetera, as time goes on. So I hope I've motivated us to be thinking about that. And there are, instead of just the word download that I gave a moment ago with some figures and images, another way to talk about a disease that's like fatty liver is to use a conceptual structure such as the adverse outcome pathway, which is, I'm going to be talking about entries housed in the wiki noted there. So the AOPs are community-sourced, expert-submitted entries that aim to catalog mechanistic information in an intentionally structured linear chain of sequential events that link across the different levels of biological organization. So again, as a I'm interested in chemicals, so of course there's a chemical property feature, but really I consider the AOP chain starting here, where we're looking at the first biological action. So something, receptor is bound. And then that will lead to a series of responses at the cell, tissue, organ, organism, and population level. So that may look like changes in signaling or protein production, all the way up through possible structural change in the population. And so what I've done is I've taken the 13 adverse outcome pathway wiki entries for fatty liver and have done my best to put those all together into this network format. And so altogether, there was nearly 120 individual events noted across those 13 AOPs, but there are ultimately a lot of overlap in that, of course. steatosis being a very popular one in the fatty liver disease AOP wiki entries. And altogether, there's just under 50 that were unique. Of those, for my purposes, I am using AOP Wiki to develop a reference list of genetic events that I can compare ROBOKOP entries to. So overall, this means that my reference list here is going to be some 34 entries um, from AOP Wiki. And so the reason I'm using AOP Wiki in the first place is because I believe that this is a great way to start improving our selection of data that will be human relevant. So this is a way to increase our human relevance of our computational models, which is my end goal. But AOPs are going to be a resource intensive effort that relies on whatever the author is motivated to add into the wiki. And so that might, or it's definitely going to limit the scope and span of what's available in the database. So it's not going to be an exhaustive reference list, but instead is meant to give an idea of if we're on the right track. So again, my research question, or my question with this project is to ask what events are missing from that AOP network? And that's me and the rest of us maybe as computational toxicologists. And so I turned to one of my lab's specialties, a tool in the knowledge graph world. And so a knowledge graph effectively is a relational database made up of entities and the relationships between those entities. And so graph structured, linking relationships between objects. We're going to put together a bunch of databases that we can then search. And the terminology here to know is that objects, entities, and nodes are all the same thing, and edges are those relationships. So if we look at this conceptual diagram, we can learn that dogs are animals, cows also animals. Cows eat herbs, which are plants. And so by looking at this toy example, another power I want to highlight of the knowledge graph is the inferential analysis. So a knowledge graph often enables structured format to then infer new connections. So we can say that cows eat herbs are plants, but we also know that cows then eat plants. And cows also are both living things and eat living things. But of course, we're not looking at cows and plants. We're looking at a biomedical database here, reasoning over biomedical objects linked in knowledge outcome pathways, or ROBOKOP. And so ROBOKOP links together 24 databases like Drug Central and linking ontologies like the BioLink model to create 11 million nodes, or those object entities, and 185 million edges or relationships are described in ROBOKOP. But before I just trust blindly the output of this database, I wanted to see that ROBOKOP can capture the expected genes and pathways that one would anticipate for fatty liver disease. So I'm using a very simple query structure of just any gene, so kind of a wild card, leading to my adverse outcome of interest, fatty liver. And so I'll show this in 3 ways. So first, looking at the gene coverage between AOP Wiki entries and ROBOKOP. Second, looking at what the top genes for ROBOKOP are and considering the scoring that went into that. And then third, I'm looking at what the top pathways emerging from ROBOKOP are, for which I'm adding a third node in my query structure. So in the validation, I feel ROBOKOP captures most of the AOP genetic entries. So those 34 we discussed before. 243 genes, or about 250, are identified by ROBOKOP. And if we consider partial matches, for which I'm considering if we have a class of genes or a family, then ROBOKOP finds 85% of those entries. And what do I mean by the partial match? There's some curation work, of course, going on here. The Adverse Outcome Pathway Wiki is explicitly structured in a way that includes particular features that are irreducible to the structure of ROBOKOP such as it is. So But not a huge issue. Just to say that if in the wiki there's a p-par, if I found a p-par A or p-par alpha, I'm considering that in the partial match scheme, and vice versa. If there's a specific and I find the general, I was considering it. But for those of us who want to be a little more strict in our definition of what matches, 70% of AOP entries were found in this ROBOKOP search. So that was one. Two, top genes from RoboCop. So here I have a histogram of the scores from those 250 some odd results in RoboCop. And the score, I should say, relies on a calculation that balances considerations related to signal and noise within the graph. So this is effectively taking into account the popularity of a node, or how many times different paths will cross through it. So how interconnected spaces are. And so the idea for this is to help refine our I think the scoring, right, to give something that is specific to the query. So that is to say that if we just zoom in on this top little bracket, we find number one, a lot of our expected AOP entries. But second, what we find too are these specific mutations that lead to a metabolic dysfunction in the liver, which is good in the sense that we confirm that we are getting the specificity with our scoring system. So something that's so well defined and linked to liver metabolic dysfunctions. That's good to see, although it may not be particularly or specifically useful for this project. I should also say that generally now fatty liver disease is considered under the umbrella of metabolically associated fatty liver disease. So it is seen as a metabolic dysfunction fundamentally. And then my third validation step was to look at what pathways are retrieved in this advanced query structure. So overall, there's over 1,900 pathways were retrieved. It's 5,000 total entries. So I'm assuming that the graph kind of ended its search expansion just at a nice even number for me. But the majority of these occur just once. So that number goes down, over half, just looking at those that repeat 2 times. And then those that seem most significant, there were only maybe 50 that were coming up quite a lot. So to visualize what I just said, most of these pathways are occurring only once. So 1,200 occur once. But there is one pathway, protein binding, that occurs 160 times, which may not be useful for our purposes. And then the second consideration here is the scoring function. So the majority of the paths are scoring well below 50%. So that's nice to have this range of scores. If I kind of turn that last figure on its side and give us the names, here we see protein binding, which again went up to 160 instances, and is maybe too generic for really to consider in this work. So if we, on the inverse, look at a score, so here I have the range of scores in the line, and then the point is the average score. What will stand out, I think, to most of us here is that the scores for our lipid process and other metabolic processes stand out. So this is good and confirms our expectations. So we get the genes that we expect and the pathways that we expect through a very simple query structure. But yet the question still lingers, what events are missing from this network? And there's us again listening along. So I found these genes to be possibly the most mechanistically important for my work. So based off of a pathway nomination, we just saw that lipid metabolic processes blew the rest out of the water. I looked at an external reactome source to consider pathways, which points in a similar but slightly different direction. And then I consider the internal results of RoboCop scoring just for any gene to fatty liver disease. And last, if I fulfill the promise of my abstract and consider how nuclear receptors contribute to fatty liver disease with the query structure pictured here, I have some nominations as well. So let's get into that. So here's the reactome visualization. So I took the genes generated by considering the bioprocesses. So I could compare bioprocesses from ROBOKOP with those noted in reactome. And I just grabbed the top some odd 10 from that tool. And so although IL-4 and 13 and 10 are coming up, what I found interesting was this adipogenesis regulation, particularly maybe in its relationship to, noting specific PPARs. And then the shout out to our disease of interest, steatosis. Felt like a good connection from these genes. And so my second nomination strategy, looking at what genes are mechanistically important, was to consider the internal scoring function, which again does take into account node popularity. And I put the top three here. But overall, there were, again, some 200 plus genes when we use this query structure, which if we consider just the top slice, there it drops to 60 genes, where we can see this inset histogram. So the majority are either very well connected or scored, and then there's this gap that we see. And then last, here's the scores of just that top subset that I showed. And so, of course, here are some knowns, and then a couple, these two I think are the unknowns that I think should be carried forward for future analysis. And my final nomination strategy was to consider what nuclear receptors to genes to fatty liver disease may show up or suggest. So I grabbed the 9 nuclear receptors, I think that are noted in AOP Wiki, and asked ROBOKOP to generate a list of genes that are in between those receptors and fatty liver disease. And I asked to look at the frequency of across all 9 of those, how many times are the same genes coming up as a strategy. And so in almost every case, PRKCA was present. I also noticed that this is SHP that's in our AOP wiki. So sometimes naming conventions can be difficult to control here. And then I wanted to star the features or I starred here the features that I think would be relevant to consider mechanistically. And so in the end here, I'm looking to identify new data and consider what mechanisms would be, or what mechanistic features would be implicated there. So I've generated or picked 3 potential pathways, and here is the total count of genes that were implicated and the particular subset that I think would be useful to consider going forward. So lipids, of course, the oxidative stress and response, and then adipogenesis pathways um, leading to fatty liver. And sorry, I think I've lost track of time, but this is my last, I think, slide, is to say that, of course, there's barriers to full adaptation. I'm sure those of you following along at home may see them as well. But one thing that stands out to me is that it's hard to meet the Bradford Hill requirements for causality with this knowledge graph. There are features that are annotated in predicates that are kind of buried in the knowledge graph through the existing user interface. But I'm looking for causality and temporality to really connect features together. And it's not a simple translation comparing AOP Wiki to our knowledge graph. And then second, and I tried to note this too, is that our internal scoring may only relate to instances of genes with a strong connection, so this mutational signature with the disease, rather than maybe elevating the importance of a specific genetic event. And so I'm looking just to bring us home with a high level perspective that this is a great cataloging of human relevant mechanistic pathways to support computational prediction of complex disease endpoints. Maybe this will have implications for some manual AOP generation, but of course also enhancing the applicability of our computational models. And now we're at the end and we're happy. And thank you all for being here today, of course, as well as to co-authors and my lab.
Thank you so much, Nyssa. That was a great presentation, really clear way to walk through what you did. I enjoyed it. I don't see any questions yet. So folks, put your questions in the Q&A, and while we're waiting, I have a question for you. I, to be honest, I had to laugh when protein binding came up because, that is a feature in so many AOPs. Like obviously, protein binding is there. Um, I sometimes work on respiratory sensitization, and I remember distinctly writing the respiratory sensitization AOP and trying to figure out if I should link that our protein binding MIE to the protein binding MIE in the liver, which was already in the wiki. Um, But I'm wondering if at the end you had the sort of three kind of buckets of pathways, and oxidative stress was one of them, and you had kind of specific genes there. And I know oxidative stress happens in a lot of different pathways. Um, is what you've done, I'm not sure if I understand, so tell me if I'm not getting it, but you have linked sort of specific genes that are involved in the oxidative stress that might be relevant to liver effects. Is that a way to kind of bring some clarity and specificity to this sort of very general event? Does that make
Yeah,
sense?
yeah. Thank you for the question, Christy. And you're absolutely right. Like this, I think organ context and the context of, is that gene active in that space, is definitely something that I think can be a complex consideration to make. So yes, in this case, those would be genes in the liver that are relevant, um, within that pathway. Yeah, exactly.
Great, cool, very cool. We have a question. Uh, have you considered machine processing of the AOP wiki data and or scoring the AOP extracted genes slash key events, for example, based on the advancement stage of the AOP or weight of evidence?
Yeah, and that's a great point too. There's more information about the AOPs that is present. So one thing I did check was to make sure that the ones I was including were were licensed, so I could take a look at them. As far as the criteria, that's very true. Whether it's been accepted or is even further, like has as part of like the six pack battery or something. Um, that is definitely something to consider. For instance, one of the 13 AOPs I looked at is just the initiating event. It looks like it was
Mm.
orphaned pretty early on. So
Yeah.
there's definitely a range of information there. So did I consider specific quality in this? Not necessarily, and because I know it's going to just be kind of a snapshot of what maybe the community thinks is valuable there. And I
Mhm.
think there was a second part of the question. Machine. Okay, so the machine processing. So there are RDFs of AOP Wiki, and I think Eva will maybe share us in the next presentation some of those. But I think what's maybe you're not specifically asking, but I am going to use as a chance to mention rule mining is so the mining of rules is an approach in the knowledge graph world that I think would be interesting. So one question I looked at answering with the AOPs was to say, okay, is it, gene related in the specific way to this next specific component? And is there a specific length to these relationships? Because if we can find a template or a rule that the AOPs follow, we can recover using those same rules,
Mhm.
the same kind of explanation from the graph. So that's not the process that I followed, but that would be a machine processing to expand beyond the AOP wiki. Yeah.
Oh, okay, thanks. And one more question. Is it possible to narrow down what cell type is relevant for those genes or pathways?
Mm. So let's see. The way that I set up my query was in ROBOKOP is to specifically define hepatic steatosis as or fatty liver as my outcome. And so by doing so, I'm limiting what genes I'm asking for to the liver specifically. So I could come up with another structure or way to structure a query that would limit to a particular organ of interest, even if I didn't know that the specific disease it was connected to. Definitely. It's a little bit tricky, but yeah, it can be done. Thanks for that.
Cool. Okay, great. Thanks a lot. Thanks again, Nyssa, and thanks for your questions. We're going to move on to Ivo, your presentation. So go ahead and share your screen.
Hello, everyone. Can you see my screen? Is
Yes.
everything looking fine? Alrighty.
Yep.
Well, first of all, thank you very much for inviting me. It's a real pleasure to present to you some of this groundbreaking work on agentic AI, and I hope this will inspire you all to integrate coding agents into your workflows and try out some of these incredible tools that have become available to us. Today, I will present something new, Talks MCP, and also a little bit of OQD, which is what he initially presented at the ASCCT conference a couple of months ago in the US, in Maryland. So one of the biggest bottlenecks that we have in modern toxicology is that we have plenty of tools, plenty of evidence that is severely fragmented in different databases. The tools are behind paywalls, they have different licenses. Basically, everything is fragmented and difficult to combine together into an actionable insight useful for next generation risk assessment. It requires manual synthesis that can take months, even years in some cases, to make sure that we have the correct applicability domain, we have the correct provenance, and this leads to an incredible bottleneck in adopting NAMs for next generation risk assessment. Now, we've all heard of ChatGPT, Claude, and the new AI chatbots that have emerged. And we have all tried them, and we've all tried asking questions, and we have realized that it's actually a black box. We often get hallucinations. We cannot trust the sources that are cited, which basically hinders adoption of these wonderful technologies. But what if we didn't use the AI to generate an answer, but we used it to synthesize data that we actually have by connecting it with different scientific tools or kernels that we have developed ourselves, such as the OECD QSR toolbox, the EPA CompDocs, PBPK models, the AOP wiki, and many other wonderful tools that we actually have. And we connect these to an AI orchestrator that will decide which tool to use when in a very explicit way, and every call is verifiable with preserved provenance, metadata, and full auditability. To try this principle, I first focused on creating OQD, which is the OECD QSR toolbox, the gold standard of QSRs and read across used by regulators worldwide. But as you all know, the interface itself is not at all user-friendly. It's quite difficult to navigate, and it takes a long time to actually come up with an answer. So what I have developed is a very simple approach that uses multi-agentic workflow interacting directly with the OECD QSR toolbox that is trusted by regulators worldwide, and split up the content into different specialist agents. And each one will look at it with a different perspective. So you have the chemical specialist, the physical properties specialist, environmental fate specialist, profiling specialist, experimental data specialist, metabolism, QSR prediction, and finally the read across. And all of these work together to come up with detailed analysis for the chunk of data that they each individually looked at, and they give everything to the synthesis agent, which creates a report that you can use with full provenance and auditability that you can use in your risk assessment. So I will just give you a quick, do you still see my screen? Just to confirm. I will just give you a quick demo of what that actually looks like. So this is the interface. You can retrieve OQD absolutely for free on GitHub. So all you have to do is copy this link, put it in one of your coding agents. In this case, it's Codex, but you can basically add a new project, copy the link, and say, help me set it up. And you are in the system, and you can run it with your own chemicals. In this case, I picked chlorpyrifos. You can pick your regulatory endpoints if you want. It's optional. In this case, I'm running all the regulatory endpoints, and you can pick, I'm running all the metabolism simulators, so that the OECD QSR toolbox is very rich in tools. You can do many things that are extremely useful. And here you can see the raw data that came out from the OECD QSR toolbox. You can see the experimental data that was retrieved, including the studies, if it's Klimisch reliability, and everything is synthesized in a beautiful report that you can directly use, and it tells you. So in this case, chlorpyrifos, what is it? It's a pesticide. It has a moderate bioaccumulation and so on. So it's checking the chemical identity, high log KOW, very lipophilic. It's no bueno. Environmental fate, environmentally persistent, especially in sediments. Also not very good. Mechanistic insight, it found acetylcholinesterase inhibition. Interesting. It also found chlorpyrifos oxon as a metabolite. And you can download here, you can download the PDF. The PDF is here. You can access it directly. And in your PDF, you have full provenance of your data, fully auditable, with everything interpreted. So it tells you, is this derived from experimental data? Is it derived from a QSR estimate? Environmental fate, mechanistic insights, data gaps. It gives you a read across strategy on how you can fill in these data gaps. What are the studies with their Klimisch reliability? So as you can see, you have an immense amount of information that is extremely useful for risk assessment with full reliability in terms of applicability domain, because it's been validated by regulators, it's been checked, it's been built by humans. So we're not asking the AI to generate these results. We're asking it to use the OECD QSR toolbox and interpret the results. We're using it as a synthesizer rather than a generator. So to go back to my presentation, the next groundbreaking thing, because this is all nice to have the OECD QSR toolbox, but it's not enough. What if we can also add PBPK models and use other open source tools like OSPsuit, like PKSim and Moby? We could use the AOP wiki to gather mechanistic evidence. We can use the EPA CompDocs dashboard, which is also very rich in tools and information. We can use AdmitLab to retrieve rapid screening of chemicals and compounds, and of course, OQD, and get a full report and a risk assessment that is reliable, auditable, at for any situation. This is how it basically works. You have your LLM client orchestrator. You can use either Gemini, Google's models, Claude from Anthropic, Codex from ChatGPT. You can use Qwen if you want to use the open source Chinese models. You can use Mistral. You can use basically, I don't want to tell you which model to use. I want to tell you you should use the models equipped with the correct tools to get the correct results. So I don't want to tell you use this model or that one. If you ask me, all of them are good if you use them correctly. And using them correctly is equipping them with the tools that we actually trust in the best way possible. So Tox MCP basically reframes LLMs for toxicology by enabling the LLMs to use all of our tools together in a fully auditable way. So it has, why do we use MCPs? MCPs are very structured. Within the tool, you can explain to the LLM, to the large language model, how it's supposed to use the tool. You can add applicability domain and safety checks. For instance, if it's outside of the applicability domain, you cannot use it. And this counts for everything, whether it's PBPK. If the PBPK model is validated for IV, you cannot use it for oral dose, for instance. You have to use it for IV, and it has policy checks directly into the MCP, which prevent you from using it outside of the applicability domain. So those are the tools that I have gathered. All of these MCPs are individually published on GitHub for free. You can try them today on the GitHub. I will show you in a bit. But basically, everything is fully auditable, and each block can be updated individually. So the idea is that everybody who is creating tools can create their own MCP and plug it in the Tox MCP dashboard to give the LLM new capabilities specific to your risk assessment question. And of course, the most important thing is transparency by design. If we are going to trust something that was generated by LLMs, we need to make sure that every interaction is fully auditable. And for this problem is solved in the Tox MCP by a cryptographic hash that traces absolutely every tool call that you made, the metadata, the version of the tool, what came out of the database, which steps led to the results that you're getting, and you get a full auditable artifact, PDF, JSON, metrics, everything fully auditable, cryptographically hashed, and replayable, so other people can also use what you've done. So this is how it actually works. You can start, does chemical X reach tissue Y, and what is the point of departure? So here you will have your agent, whether it's Codex, Claude, or Qwen, or whatever you're using, Gemini, up to you, whichever is your favorite. I personally use all of them for different reasons. I think all of them have their strengths and weaknesses, but they all work exceptionally good if you plug them in with the right tools. And those are the tools here. So it will use the comptox to resolve the identity. It will use PBPK to derive a Cmax. It will find mechanistically relevant events. And it will use the OQT and everything into a fully auditable report. So what is the difference between OQT and Tox MCP? Is that OQT is a single tool which is focused on the OECD QSR toolbox. It is also auditable, and you can run it. But the Tox MCP is an actual ecosystem. It's a multi-tool federation that allows you to combine different tools together to come up with actually strong conclusions. And if you think about it, what do we need for regulatory acceptance We need standardization, traceability, and of course, we also need efficiency, because if it takes 1 year to come up with a conclusion, well, that's not going to be useful for testing the 15,000 new chemicals that are registered with a new CAS number every single day. And of course, you have to understand what you're doing to ask the right questions. So you need to be trained in risk assessment. You need to be able to ask the right questions to get the best answers. Of course, anybody can ask questions to ChatGPT, but when you're actually doing risk assessment, you need to be trained in asking the right questions. For instance, how is the exposure done? Who are the people at risk, etc. So let me show you a quick demo of Tox MCP itself, because it's absolutely mind-blowing. I'm just going to take an example from Nyssa, who was just presenting something about the AOP MCP, no, about the AOP wiki and the knowledge graphs. So knowledge graphs is not something that the MCPs can do, but for instance, maybe Nyssa can make an MCP and integrate it together with the AOP MCP, and then we can actually create what she did, the knowledge graphs for any AOP. So I just ask a question. Let's try fatty liver disease and see what you get from the AOP MCP. So here, what it did, it pulled 12 fatty liver AOPs, 3 non-alcoholic fatty liver disease, 9 non-alcoholic fatty liver disease written differently. And for each one, it gives you the confidence profile. And the confidence profile, how is it derived? It's derived based on the OECD guidelines for AOP building and confidence. So in this case, we have low confidence. In this case, we have moderate confidence. But maybe Nyssa can tell us if this makes sense or not. I think it does. And the beauty of it is that you can say, okay, let's now retrieve the chemicals that are associated with these AOPs. So it ran the EPA CompTox and it pulled chemicals that actually are associated with assays, with liver assays that map to fatty liver AOP. So here you have the chemicals. In this case, we have PFAS. It's a famously known direct match PPAR lipid dysregulation leading to steratosis. 25.6 active assays. You have the summary signal, how many records, NOAEL, and so on. In this case, I don't have a PBPK model, so you do need to have a PBPK model pre-built for the MCP to work. So you can basically take any PBPK model that exists, developed by SQ Labs, for instance, or by researchers using Berkeley Madonna or whichever software they're using, and you can run it here. So I will show you what the PBPK MCP looks like. In this case, I'm using cisplatin, which is a PBPK developed during VHP for Safety project by Kiri, who is a PhD student at Wageningen University. And she has done an incredible job developing cisplatin PBPK. And here we are running it. So it took in the PBPK and it outputted systemic components. How does it distribute in plasma, in kidney? What are the different microcompartments? How does it get eliminated after how many hours? You can run population simulation and take, for instance, in this case, we take 60 individuals from 35 kg to 140 kg. How does it differ? Because of course, each individual parameter can affect the distribution of the chemical. So if you combine the different MCPs, you can actually solve many different questions. So let's look at another example, which is chlorpyrifos. I showed you OQD with chlorpyrifos right before. And here I start the question with, can you use CompTox and retrieve how chlorpyrifos was used? So it uses the EPA CompTox, it pulls chlorpyrifos, identifies it. It found 266 uses, of which 260 are insecticides. You can find that there's also the 6 minor labels, degrades, pesticide variants. There are some impurities as well. Product use, pesticides, laboratory supplies, agriculture. So basically, you can use this information. You see, it was used from 94 to 23. You can use this information to come up with a realistic exposure scenario. So if you're asking, can you use all the MCPs and tell me if chlorpyrifos can actually reach the brain tissue and cause long-term Parkinson's disease? Now, that is a very difficult question to answer um, um, in 10 minutes, but I will show you now that it's actually possible. So what we get from the EPA CompTox is the physicochemical TK signals, bioactivity burden is substantial. Then we use the AOP MCP. It identified acetylcholinesterase inhibition, which is directly relevant for chlorpyrifos. It found neuroinflammation, cell injury, Parkinson's relevant diseases. Actually, one of those AOPs was developed during VHP for safety. So this is a case study that we've been carefully working on. From the OECD QSR toolbox, we ran some QSRs and we can see that the blood-brain barrier shows good permeability, which is no bueno. Oral absorption, it's highly absorbed, and it's a high Cramer class. And it also has an oxon metabolite, poor permeability. But luckily, the oxon metabolite has worse permeability than the parent compound. In this case, I don't have a PBPK model for chlorpyrifos, so I couldn't use that MCP. You have to have a valid PBPK if you want to use it. You cannot just use a random PBPK. Or you can create a simulation, like a 2-compartment simulation, but this is not scientifically valid. And it knows that. So it did make it, and you can ask it to generate figures for you. So let's create a figure of what we have and what we're missing. In our case, chlorpyrifos, we have the exposure evidence from CompTox. OQD gives information on central nervous system penetration. We have information on metabolites, acetylcholinesterase mechanistic, neuropathway, Parkinson's, AOP availability. So what we're lacking really is the PBPK model here. Even though this made a nonsense 2-compartment model, it's still lacking in terms of evidence. So this is one quick scenario that I'm showing you here. But of course,
Ivo,
yeah,
we have quite a lot of questions in the
I,
Q&A, so I wonder if we want to try to
yeah,
finish up and move
I
to.
will cut it right now. Um, um, and I'm happy to answer questions. And I would encourage everybody to actually try these tools on GitHub. They are all available for you on this repo, Talks MCP. Please star it to make it more visible so more people can see it, use it, and contribute. But basically, everything is explained in detail how you can use these tools. The easiest way is to just copy the link here, paste it directly into your coding agent, whichever you're using, and it will set it up for you. But of course, you need the API key for the CompTox. You need access to a real OECD QSR toolbox. So you cannot use these tools if you don't actually have these tools installed locally. And for every tool, there's a separate repo. And for every repo, you have an explanation of the architecture, how it was built, why it was built. You have all the information of what it can do, limitations, setup, and so on. So thank you very much. It was an absolute honor to present this tool, and I'm looking forward to hear what you all think. This is fully open source, so you can go after closed source tools, but I believe that this is a new age where we have to share everything that we're doing if we actually want to make a better world. Now is the time to share what we got and come together to make this world better because the clock is ticking for us all and we finally have the technology to do it. So I hope this is also inspiring for you to try out and also share what you're doing with the community. So thank you very much. I'm very happy to hear what questions you have from everybody, and hit me up on GitHub, on LinkedIn. You can also email me on inforar.ai if you would like to work together on solving some of the most difficult problems of our times. I'm always happy to engage and help you out. Thank you again one more time for the invitation for this talk, and looking forward to hear what you think.
Yeah, thank you very much for the talk and the demo. We have quite a few questions. Just to note for everybody, there were a few questions about asking for the link. So we have the link to both OQT and Talks MCP in the chat. So you'll be able to get them there. So the first question is actually a question that I had as well. When you mentioned, that you can run the OQT with whichever AI model you'd like, ChatGPT versus, or even 5.4 versus 5.0. Have you run them across different models so that the output is consistent? Have you checked that?
Absolutely. I've actually tried it with the weakest models. When I was building these tools, I was only using 4.1 nano because it was the cheapest, and I was doing a lot of tests. And basically, the goal was to make sure that it can work with any tool, with any AI model, including the open source models such as
Mhm.
Qwen 3.5 that recently came out, and it's an absolute banger of a model, tiny and mighty. So it can work with practically any, because you're using the AI as a synthesizer rather than a generator. So the generative power is not as important in this case because
Mhm.
you're using
Okay.
the actual tools that we've built and validated ourselves, if
Okay.
that makes.
Yeah. Can you plug two or more stressors into the tool to predict interactions?
I have not tried that, but that's a very good question. We could try it now if you want, but
We
we
have a
have.
lot of questions to get through, so let's just answer them
Okay.
quickly and we'll
Great.
you
Please
can
try it.
try
So this is
and let
all
us know.
new. I mean, I'm also discovering the capabilities of what is possible, and I'm constantly developing these MCPs, trying to gather feedback from everybody. So every feedback is appreciated. Would love to hear how you use it, what use cases you
Sure.
discover, and I'm also discovering this with you, right? So it's very new technology.
It's
We have to
exciting.
make the most of it.
So the next question is, can you tell us more about how you made your specialist to be a specialist? So what is the approach behind what you built?
So the approach to make the specialists is to feed them only the information that they need to come up with the answer, and also prompting. So each specialist has a very specific prompt. You are looking at, for instance, environmental fate. Your specialty is, your focus is environmental planetary protection. You're gonna focus only on environmental parameters. Then we have another one. Your specialty is metabolites. So the one that is specialized in metabolites will only get access to the tool of metabolism prediction. And it's very it's very good only in that direction. And then the synthesizer takes the input of everybody together to come up with the report, if that makes sense. So each individual agent is specifically built for a specific task. And if you go on the GitHub repo, if you copy the link, put it in your coding agent and ask, can you explain how these agents work, it will actually tell you and walk you through, so you can also build your own agents. Because that's the beauty of this system, is that you can also build your own and integrate it in the framework. So I can also use your tools just like you can use my tools, and in the end, the world is better, right?
Mhm. So next question, have you thought about how this tool will be maintained in the long run and where that sort of functional support will come from?
So in this case, it's 5 tools. Each tool, each MCP is a separate Docker container
Mhm.
that can be separately updated and maintained. So as we add new tools to the Tox MCP framework, um, you can update each element individually if needed be. Like for instance, yesterday I updated the AOP MCP. Today I updated the PBPK MCP, and I added the support for Berkeley Madonna and R. Whereas before it was only PKSim and Moby from SQ Labs. So I'm personally boosting these MCPs uh, regularly. When people tell me I would like to have this new ability, I will try to add it myself. But it's also fully open source. So I'm hoping that the community as well will maintain some of them, add new MCPs to the to the framework. So MCPs are very structured, but there's another thing called skills, and I'm hoping as this technology matures, people will develop the skills and improve the MCP, so then we can really make powerful risk assessment together. That's the idea.
Okay. So I think this question is specifically about the OQT. Um, and it's asking in a simple term, what's the difference from a report generated using just the OECD QSAR toolbox report generator function?
Um, well, I think the main difference is the depth that you get, because this is interpreting every single parameter that the toolbox actually has. So if you're an experienced risk assessor, you probably already know if log KOW is low, is probably bad. Um, but it helps you collect absolutely every evidence with a different perspective, because each specialist agent will look at the data from a different perspective and provide useful information. And at the end, it will give you a really good read across strategy that you can actually use, read across justification and strategy that you can actually use based on the data itself. So it is kind of different. It's more, you get more detail, and you can do it very quickly. Whereas if you go through the original OECD QSR toolbox interface, it's going to take you way longer unless you're very experienced. Um, so yeah, that's the main difference.
Okay, great. Um, and I think the answer is ready to use, but the question is just to confirm, are these tools ready to use AI models in web servers, or do you need coding skills to use them?
No, you absolutely don't need coding skills. Actually, coding is over. Nobody's doing coding anymore. All you need to do is install Codex from ChatGPT, or Claude Code from Anthropic, or Quen Code, or Gemini CLI. You have many options. But if you're not using any coding agents, please trust me and try them out. It will really change your life. You can do so many things with these tools. They can create code for you. Now what matters is programming. But we all know we're all born with the skill of programming. Every morning, you know what you need to do. You open your eyes, you get out of bed, you go to the toilet, drop your pants down, you take a pee. You do it every single day. So we are natural programmers. But if I ask you what is the first thing you do in the morning, you're gonna tell me, yeah, I go to the toilet. But no, first thing is you open your eyes, get out of bed, So all of these things are abstracted in our head. And now the same principle can be applied to programming by using the agents to abstract a lot of the small details, and we get straight to the point of what we actually want to create and achieve. So please try out the coding agents. It will really help you. And it will help me as well, and it will help all of us because we all have different dreams, and we're all creative in unique ways. That's all I'm trying to say.
But if you really don't want to code, you don't have to. That's the moral of the story,
No,
right?
actually, I use speech. Personally, I only talk. I
Okay.
use this.
Hmm.
I click to dictate, and then I'm just talking, and then I say, okay, what else can you pull out from the database? And then I click send. And as you can see, it already used speech. So the future of programming is actually speech. At least for me, I spend a lot of time on my phone recording what I'm saying, and this is how I build practically everything. You see, it's already doing it. And it's writing the code. It's running the code in real time. So
Okay.
no, you don't need to learn how to code anymore.
Let's get
What-
back to our questions, though.
Yeah.
Just a couple
Yeah.
more. Um, all right. Have you faced any issues with chemicals that have a lot of data coming from any of the databases that you're coupling with the LLM? So if there's sort of like an overload of data, I think, or have you found issues with the orchestrator summarizing findings, for example, neglecting relevant information?
To be very honest with you, I haven't tried it with many chemicals. I mostly focused on the case studies that we have with the VHP for Safety project because we have entire teams trying to build the regulatory workflow and risk assessment approach for these case studies. So it kind of helps me get an idea in which direction I need to go. But I guess I haven't tried it with thousands of chemicals, and that may be something that I should do. But I have not so far had such an issue. I think the models have a big memory context. The models that I'm using have 400,000 to 1 million tokens, which is about 600 pages. So unless there's 600 pages of text, unless you have a Bible written on your chemical, you probably won't have any issues like that.
Okay, great.
Most issues are from lack of data. Uh,
Right. Yeah, we know that. Um, Actually, there is a question for Nyssa. So, Ivo, I'm going to let you look at some of the questions in here and see if you want to type a quick. I think one of them you answered, but maybe you want to be sure, the one from Logan. But Nyssa, let me read this question for you. It's a follow-up to the cell type, relevant cell type question. And so it's, for example, adipogenesis refers to the differentiation of precursor cells into mature adipocytes and adipose tissue. In contrast, fatty liver is mainly characterized by triglyceride accumulation in hepatocytes rather than the formation of new fat cells. It seems unexpected to observe adipogenesis pathway in liver data. So it would be helpful to understand whether the signal reflects a specific liver cell population or a limitation of the pathway annotation.
Yeah,
Oh,
and oh, sorry, was there more? Or
nope, go ahead.
okay, thanks. Yeah, and so thank you for your clarification to the question. I was starting to type up my response, but I think fundamentally, maybe it would be to say that Reactome is nominating adipogenesis based off of the gene list generated by RoboCop. So I think maybe they're like the specific liver context that the gene list was generated in isn't necessarily accounted for with in Reactome. So I think it's responding to whatever that gene list is. But I have two more thoughts related to that. First, maybe this point means that it won't be necessarily as useful for my computational models because my computational models are relying on in vitro data. So maybe adipogenesis is because I know that there's connection to like brown fat formation and liver steatosis. So like that the metabolic dysregulation, even though fatty liver is a manifestation of it, it's still a system-wide issue or phenomenon. And so um, I yeah, so the human relevance, but so maybe it's relevant, but not in, I wouldn't necessarily see that data coming out of this in vitro studies. So um, the pathway generalization that identified, oh gosh, adipogenesis is something that's agnostic of the tissues. Whereas my like geneless generation would, I think, be liver specific. Um,
Okay.
yeah, but definitely something to take into account. Thanks.
Yeah, sure. Thanks, Nyssa. We have maybe two more quick questions for you, Ivo. So one of them is, what level of risk is inherited when using the MCP summary prediction for chemical tox assessment, which relies on read across and prediction of QSAR in the absence of experimental data?
It is the same risk that you have when you're using the OECD QSR toolbox and any other tool that you use in your workflow. So the risk is in the hands of the risk assessor and the questions that you ask and how you use the information that you get from your tools. Of course, there is a risk, but these tools are for risk assessors. They're not tools for my mom, for instance. She can also ask questions, of course, but she cannot make conclusions.
Right.
And there is risk. It's the same risk that comes with the tools that we have. And I'm not saying that our tools are perfect. There's still a lot of room for improvement, of course.
Right, of course. And another question is, there remains the challenge of consensus among decision makers. Are benchmarks being developed to compare outcomes with numerical results or from different agent results? I see we're already typing an answer, so what do you...
I was about to say that I do have my personal benchmarks that I use to test my personal agents, but these benchmarks I am not ready to publish yet. I still have some work that I need to do on that. And I use it for my personal use. So in my benchmarks, I have a lot of tests, not only toxicology related, but also coding related, also retrieval related, how successfully it can make tool calls, and so on. So I do have my personal benchmarks, and I would encourage everybody to develop their own if you want to trust that there is development in the LLM field going on, because as soon as there's a new model that comes out, how can you be sure that it's actually better if you don't have your own benchmarks that you
Mhm.
can trust? So yes, there are benchmarks, um, and I will share my benchmarks when I'm ready, but I'm still not ready. As you know, I'm open sourcing everything. My principle is to share as much as I can with the community, but I don't want to share something that is half baked. Like it
Makes sense.
needs to be ready.
All right, one last question. Can you share some light on risk of prompt injection issue?
Um, there is a risk of prompt injection as that is something inherent to any LLMs. Um, in my case, the risk of injection is mitigated because I'm using the trusted tools. I'm not using internet to answer
Mhm.
my questions. I'm not asking my AI agents to search Google and go on Reddit to pull information about my toxicology endpoints. I'm asking it to use the official OECD QSR toolbox, which I assume doesn't have prompt injections inside because it was developed pre-prompt injection era. But of course, there is a risk that a bad actor might inject something malicious in the EPA CompTox dashboard, and then you end up deleting everything that you have. And you also have to sandbox. Personally, I sandbox my agents. In this case, I did not. As you can see here, I gave it full access. But if you use a sandbox, you are generally more protected. And the only reason why I give it full access is because I don't give it access to the internet in this case. I'm just using the MCP tools
Mhm.
I build myself, and I trust that it's prompt injection free. But yes, we should all be aware of these risks. These risks are real. And this is something that I would like to tackle with my company. When it comes to making putting these tools as at an industrial grade, you need to make sure that you're protected from this. And this is something I can offer as a service, but it's not something I can guarantee when you use it yourselves because I don't know how you will use these tools, if that makes sense.
Certainly. Okay, well, really appreciate your demonstration, your presentation. Nyssa, also really appreciate hearing from you and hearing about your work. So Ms. Tucker, Dr. Djidrovski, really appreciate having you. Thanks everyone for joining us and for asking all of your excellent questions. And we'll see you again sometime soon. Bye-bye.
Thanks.
One more
Bye.
reminder, please
Oh,
start a GitHub to make sure more people see it and
Okay,
use it. And yeah, I appreciate you all. Thank you.
thank you. Take care. Bye-bye.
Bye-bye.